Hi everyone. I hope I'm audible. So let me just check once. Yeah. Okay. So today we are going to discuss about the phases of cell cycle. Okay. And let's see what all changes that we can see in a phases of cell cycle. 1 second. All right. Okay, so before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE class 9 and 10 biology. Okay, and the Telegram channel, that is Unacademy ICSE and ISC boards. So this link you can find in the description box. Okay, so go to the description box and find the different links which are there. So one link is basically special class link you can find, and also the Telegram link of Unacademy ICSE and ISC boards you can find, and the community link you can find. Okay, so go join these channels and also click on the special class link. So I'll tell you what is the benefit of special class. So you can learn from your favorite educator for free. Okay, so different educators will have their own courses, they'll have their own classes. So you will have access to these classes because these are absolutely free, and you can also engage with educators via polls. Okay, so that means we will be conducting a quiz. So you will have you will get to know once the quiz is done. So we will be sharing the leaderboard so that you get to know your rank and you can compete with your friends as well. So clear your doubts in the live special class. So if there are more children, okay, so usually chances that your question might get lost because of the comments that keep on coming. So there is a separate question tab where you can type in your question and send it to the educator during a live session, and they'll be clearing your doubt. Okay, so for the top special classes you can visit the link in the description. Now Unacademy subscription features, let me just check once. Yeah, so Unacademy subscription features, so you can learn live from the comfort of your home. Okay, and unlimited access to all the courses. So different educators will come up with their own course, so you will have access to all the courses once you take the subscription. Then top educators of India are going to take the class in one platform, that is Unacademy platform. Regular doubt clearing session will be done. So after about every 3 sessions, the 4th session will be your doubt clearing session. So any doubts you have related to the topics that are previously covered, so you can clear your doubt in a doubt clearing session. Then exhaustive coverage of a syllabus, that means complete syllabus will be covered. So we will be following the reference books also, so you can you can practice with us. So we will discuss about the diagram based questions like 3 marks question, 5 marks question. So each and every question will be discussed. So that means this is going to be helpful for your exam. Okay, then mentorship and guidance will be given, study material is given, the practice tests and live test series, and batch courses. So you will have access to all of this. Now batch course, when we mention, you have a book. Okay, 1 second. So Selena book, right? Yeah, so for textbook anyway, you best book is the Selena book that you can refer. Okay, so this is the best book you can go with. For references, workbook I'm talking about, like you can go with the Together book. So this is just for your practice, you can understand what type of questions can be there, like 2 marks question, 3 marks question. So you will more you practice, you will have an ideology like what type of questions that you might get for your board exam. So reference books are basically you can go with Together with Oswald publication, Goyal publication. Okay, so all these are good books, so you can refer them as well. Okay, but Selena is textbook, which is the best one. So if you are perfect with that, then there is no need of any other book, but you need to know the type of question. So for that you can refer previous year questions also. Okay. And yeah, so batch courses, once you once you take the subscription, so you will have access to the batches where the mode of delivery can be English, or it can be English as well as the Hindi. So you will have access to all the batches once you take the subscription. Okay. And for class 10 you can just try with the 3 months of subscription as well. So now this is the very beginning for your class 10, so you can go, I suggest you all to go with the 12 months of subscription as so this is the very beginning, and with the referral code, so this is the code Vani Kari. So with this referral code you will be getting 10% discount on the subscription. So what you can do is, so basically for 12 months it is 15k. Okay, when you apply a code Vani Kari, so this is gonna get reduced to 13.5k. okay, so you can go with the subscription for 12 months since this is the very beginning. Okay, so let's get started with the cell cycle. So today I'm going to discuss about the cell cycle here. Okay, and let's see the phases of the cell cycle. Yes, hi Bhaibhav, so do you have any idea like what exactly is cell cycle, why does it occur, or any idea related to cell cycle, any of you? So why there is a cell cycle, why cell cycle has to occur, and what are the types of divisions that we can see or phases we can see in a cell cycle. So basically when we mention a cell cycle, so this is a preparation stage. Okay, so this is a preparation stage just before the cell division occurs. So there are a lot of changes that are going to occur. So cell cycle we can see in two stages. Okay, so let me just explain you this. So one second. Yeah, now this, let's say this is now a cell cycle. Okay, so in cell cycle basically you can see two phases. Okay, so one is M phase, so there you can find a M phase, and other than that this whole phase what you see, so this is completely a interphase. Okay, so interphase is the biggest phase where this phase is actually a preparation phase. Okay, so preparation for what? Preparation for the next stage, that is M phase. Okay, so what exactly is being prepared here, like just before in our homes and all, so when the cooking has to be done, so what we do first, so first we are going to keep the raw materials ready, like so we cut the vegetables. Okay, and we wash the rice. Okay, so we make this we need the dough. Okay, so all these changes are done, right? So then we go for the cooking, so that we on the stove and we do the cooking. So it is just like that. Interphase is basically keeping all the raw materials required for the cell division ready. Okay, so what are those raw materials, like cell organelles are gonna multiply. Okay, so cell organelles are gonna increase, or we can say replication. So DNA is going to replicate, the protein production is gonna increase, the RNA synthesis is gonna increase, so there is ATP synthesis is going to increase. So proteins, that is plasma membrane proteins, are kept ready, tubulin proteins are kept ready. So basically interphase is just keeping all the raw materials ready so that the successful completion of cell division can occur without any problem. So this is now in M phase you will study many different things. So first we will see this interphase, which is a part of a cell cycle itself. Okay, for growth regeneration, yes, so that is mainly cell division occurs, so that is for growth, in order to replace the old and dead cell. So for that there is a cell division that we can see. So before the cell division, so there is a preparation, a lot of preparation has to be done, so that is the interphase. So let's see this one. Okay, so now definition: the series of changes occurring in a cell leading to the division of a parent cell into two cells. So just before this part occurs, okay, so just before this part occurs, so series of changes are occurring. So what is that series of changes occurring? So basically cell organelles will replicate, okay, so replication of cell organelle, replication of DNA, okay, so these are the main things that are required, and replication of, like, production of protein can also be seen, and along with that large amount of ATP, energy currency, is required for, so that the cell can divide easily. Okay, so for cell to divide, usually ATP is required, so large amount of ATP is required, so ATP is required. Now RNA, when we mention, so RNA is also required. I'll tell you how. Okay, so I'll just explain you that now. Okay, I'll explain it to you here. Okay, so now DNA is there already in a cell. DNA is there, okay, and this DNA has to produce a RNA, okay, so that we call as a central dogma. So basically transcription process will occur, and then RNA is going to produce a protein. So we say that in cell cycle the RNA amount is increasing, the protein amount is increasing. So that is because DNA is transcribing, or DNA is producing a RNA, so that the required amount of protein can be produced. So what is this protein? Now in order to form a plasma membrane, you require a lot of protein, like plasma membrane. So since the cell division has to occur, so plasma membrane is basically made of lipoproteins, okay, so that means protein is required. Now spindle fibers, we say, right? So for spindle fiber formation, so there is a tubulin protein that is required, okay, so tubulin protein is being formed because spindle fiber has to be formed in the M phase of the cell cycle, okay, so M phase when we mention, so that is mitotic phase. So spindle fiber formation we can see starting from the which stage? Prophase itself, okay, so for that protein is required. Now one more thing, before that we require another set of protein here. So what protein is that? So now we will see the cell cycle. So there is a phase called as S phase, okay, so in S phase DNA replication occurs, okay, so DNA replication we can see in a S phase. So for DNA replication to occur in a S phase, what is required? So there are lot of enzymes are required for this DNA to be copied, okay, so let's say these are the parent strands, okay, so this is now a parent strand. So we need to form a replication, that means semi-conservative type of replication we can see. So that is with respect to the DNA that is already there. So now here you can see in this one, so these two were parental strands, okay, so here you can see semi-conservative. So this is one copy produced, this is another copy produced. So one of the strand is old strand, the other strand is basically a new strand. So here in this case, this is new strand, this is old strand. So what has happened here? The DNA which was there earlier, so that got separated, okay, so DNA strands got separated, so new strand synthesis began there itself, okay, so new DNA strands started to synthesize. So now in order to synthesize this new DNA strand, okay, what is required? So the required thing is basically a nucleotide, okay, so nucleotides are required. Then the enzyme, so enzyme is basically protein in nature, so protein has to be synthesized in the cell cycle, so protein is required, okay, and for the formation again ATP is required. So protein formation will occur only after the RNA formation. So nucleotide, RNA, protein, ATP, so different things are required for the S phase to occur, okay, so we will discuss each of these stages one by one, so you will clearly understand how exactly these are going to play a role in synthesizing a new DNA and how exactly the cell is getting ready for the cell division. So let's see that. Okay, so now before I discuss with that, one second, so let me just explain you this. Okay, so now here human cell, okay, so human cell for the cell cycle, so this will take 24 hours. Now for this one cell that we have to divide it, like to completely undergo cell division, so we take 24 hours. Okay, so not just some minutes, it is 24 hours. Now in case of a yeast cell, so it is 90 minutes. Okay, so 90 minutes. So there is one particular bacteria called as Escherichia coli. Okay, so this takes a time of 20 minutes for the cell to divide. So here in case of human, it is 24 hours. So long time is required here. Okay. So now phases of cell cycle is the interphase is there, and you can also find a mitotic phase, or we can say M phase in general. Okay. So duration of the interphase, so more than 95% of the cell cycle is interphase itself. So you can imagine, so let me just explain it to you here. Okay, so now let's say now here okay, one second, I'll just take the pen. Yeah, so now this is a cell cycle if I say. Okay, so here in this case 95% is interphase, only 5% is M phase, where you study prophase, metaphase, anaphase and telophase. So that 4 stages along with the cytokinesis what you are studying. So that is just the 5% what you study so much in detail. Okay, but here the one that you are studying, that is the interphase, is way too important, because this is just you will understand like what exactly preparation is done. So now this is a M phase, which is just a 5% of whole of the cell cycle. Now this stage is basically interphase, which is about 95%. Okay, so now interphase is again divided into 3 phases. So let's see this one. So one is basically a G1 phase. Okay, so G1 phase we can also call it as a gap 1 phase or growth 1. Okay, so in your Selena it is given as growth phase, or it can also be called as a gap. Okay, so growth 1 phase or gap 1 phase. Next phase is actually the S phase. So S phase is what? So this is a synthesis phase. Okay, so this is a synthesis phase where the DNA is being synthesized. So there are lot of changes you can see in G1 phase, lot of changes you will see in S phase, lot of changes you will see in G2 phase, and only after this whole interphase is complete, okay, the cell is now undergoing for the M phase. Okay, are you all understanding till here? Is it clear till here? You want me to repeat any part? I'll explain you like what is going to happen in G1 phase, S phase and G2 phase. Okay, so I'll just explain that. Now you can see here, so this one is, so those diagrams of anaphase, metaphase, prophase. So do we need to practice? Yes, that's very important. So yes, I think today's, yeah, so yesterday's session, okay, so that is IPL, you can watch ICSE Premier League, which I have taken on YouTube itself. So you can watch that session. So that is very important. So you will see lot of previous year question papers based on these, all these prophase, metaphase, anaphase and telophase. So you need to remember each and every part, like each and every point, and how exactly the diagram is there. Okay, so lot of previous year question papers, I mean not question paper, the previous year question paper you can refer. So that means it is very important. Okay, and yes, so G2 phase is basically a growth 2 phase. Okay, so let's see growth 1 phase first. Okay, so cell cycle, so this is going to be divided into interphase and then M phase or a dividing phase. So gap 1, yes, and gap 2, or we can, instead of gap, you can also use the term growth. Okay, so it's better you all use growth, because in Selena it is mentioned as growth, so go with the growth phase. Okay, so growth you can use, and in M phase basically cytokinesis as well as karyokinesis we can see. So this is karyokinesis. Okay, so this one is karyokinesis, that is division of nucleus will occur in 4 stages. One is prophase, metaphase, anaphase and telophase. The other one is cytokinesis. Okay, So let's see this one. So cell division, so let's see that. So I'll just explain you the first stage, that is G1 phase. Okay, so that is growth phase or growth 1 phase. So what exactly is gonna occur in growth phase? So in growth phase you can see that the cell organelles will replicate. Okay, so you can find mitochondria is there, then there is endoplasmic reticulum. Okay, so different things are there. So these cell organelles are going to replicate. So that is in the growth 1 phase. Okay, then what exactly is this one? So why exactly this is required? So when the cell division occurs, so these these cell organelles are gonna be helpful. So now other than that, you can also find the cell organelles are anyway they are replicating. So along with that, you can also find that the RNA synthesis is gonna increase. So why exactly RNA synthesis increases? So let's write this completely. So RNA synthesis increases. So why exactly? Because DNA is already there. Okay, and we need to produce. So from DNA only RNA production occurs, from RNA only protein production occurs. So this we call as a central dogma. Okay, so this is called as a central dogma. That means the DNA is giving rise to RNA, RNA is giving rise to protein. So process here is transcription, the process here is translation. Okay, so different processes are there. Now, why exactly RNA synthesis? RNA synthesis is increasing so that the protein production can increase. And why protein is required here now? So protein is required because proteins are enzymes which you can see in the cell replication. So those are protein in nature. Okay, so this is just like G1 phase is just a preparation stage. Okay, so this is a preparation stage for which one? Preparation for the S phase is happening in the G1 phase. So what is that preparation? So as the RNA synthesis occurs, so protein synthesis occurs, so enzymes required for what? Enzymes required for the DNA replication. So those are kept ready here in the G1 phase. Okay, so enzymes required for the DNA replication, so all those are kept ready here in the G1 phase, so that yes phase, so that in the yes phase what is going to happen? The easy replication of DNA is going to occur because all the enzymes which are protein in nature, so those are kept ready. Okay, so along with that, so you can also see there is a ATP synthesis which is going to increase. So why exactly ATP synthesis is required? Now so basically DNA replication we can see. Okay, I'll just explain it to you here. Okay, so now let's say this is an old strand of a DNA. Okay, and a new strand has to be formed, so that is semi-conservative. So we say that DNA replication is semi-conservative. So what is the meaning of semi-conservative? The old parental strand is conserved and new parental strand is being synthesized. Okay, so now this is a new, not new parental, so new strand is being synthesized. Okay, so here also old strand is there, so new strand is being synthesized. So what I am drawing as line here is, so these are basically just the nucleotides. Okay, so these nucleotides, what are them? So these nucleotides have to be joined. Okay, so here in the G1 phase, so that is in the G1 phase, the nucleotides are also kept ready. Okay, so that is the raw material. So nucleotides are kept ready so that the S phase, the DNA synthesis is gonna occur easily. So with respect to that, we can say here, let's say this. So now in order to join these nucleotide, okay, so there is a requirement of the ATP. So different enzymes are required, ATP is required. So this basically new strand is being synthesized. So that means now this one is a new strand. For joining these nucleotides, okay, so for joining all these nucleotides, so there is a requirement of ATP, so there is a requirement of an enzyme. So for that reason, the protein synthesis, RNA synthesis and ATP synthesis is going to increase. So now this strand is a new one. Okay, so here this one was an old strand. Now again, in order to join all these nucleotides together, so there is an enzyme requirement, there is an energy requirement. Okay, so for that again ATP and enzyme is required. So now here in this phase, that is G1 phase, so we can say that hi Anita, hi Samriddhi. What is RNA? So I'll just say that. Okay, so RNA is, one second. Yep. So RNA is basically, you know, the ribonucleic acid. So that means, so it is quite difficult, like, you know, not difficult, sorry, different from the DNA. That is because RNA is having a ribose sugar. Okay, so sugar which is there in RNA is ribose sugar, whereas in case of DNA the sugar is basically deoxyribose. Okay, so rest of the things, you know, it is somewhat similar. So some changes we can see with respect to bases. So instead of a thymine which is there in a DNA, so we can find a uracil in case of a RNA. And the sugar which is there, so in case of RNA it is a ribose sugar, whereas in case of a DNA it is a deoxyribose sugar. So structurally, so you can find certain changes you can find in a DNA and RNA. So DNA itself produces a RNA and RNA itself produces a protein. So now for all these semi-conservative, that means now old strand is maintained and, you know, new strand is basically being formed. So that means only half of the parental strand is conserved. Okay, so this is conserved. The new strand is actually, you know, the formation will occur. So here, you know, the old parental strand is conserved. So that means it is not complete conservation. So it is the semi-conservation. Okay, so 2 copies of the DNA are formed here. So that is because of semi-conservative replication we can see. Okay, so here in the G1 phase we can see that, so there is basically, you know, all the raw materials which are required for the S phase, so this is for the S phase, are kept ready here in the G1 phase. Okay, so moving on to the next one, so you can see here interphase. So this is basically a preparation stage itself. So let's see the gap 2 phase. So before that, let's see this one. So gap 1, or we can also call it as the growth 1 phase. So this is growth 1 phase also. Okay, so this is an interval between the mitosis and initiation of the DNA replication. So that is S phase, between the S phase and between the M phase, whatever gap is this, so that is a G1 phase. Okay, now cell is going to grow continuously. So here in G1 phase, what exactly happens? So we can see a growth of a cell. Okay, so growth of a cell, so that is with respect to all these changes that are going to be occur. Okay, so now this G1 phase is, you know, it is also called as a growth 1 phase or gap 1 phase, and we can see that there is a replication of few organelles. So like mitochondria is being you know, multiplied, endoplasmic reticulum can get multiplied. Okay, so then cell in a metabolically active state we can find. So just a second. Yeah, so cell here we can find, so cell in a metabolically active state. Okay, so that means it is continuously going on producing RNA, it is going on producing DNA, then ATP synthesis is occurring. Okay, so all these changes can see, and here there is no DNA replication. So DNA is not produced in this stage. Okay, so that is about the, one second. Yeah. So that is about the DNA replication which is not occurring here. So now in the S phase, so what exactly happens in the S phase? So you know that in G1 phase all the raw materials were kept ready. In S phase, so DNA replication we can see. So DNA replication, So that is what type of replication it is? So that is basically DNA synthesis. So yes phase, the name is yes phase because synthesis is occurring. So synthesis of what? Synthesis of DNA we can see here. So that is by a replication, which is actually a semi-conservative type of a replication. Okay, so this is a semi-conservative type of replication. Now the other cases, the centrioles are also actually replicating here. Okay, so centriole replication also we can see here. That means one of the cell organelle which is required for the cell division to occur, so that is also multiplied here itself in the yes phase. So yes phase, this is also known as the synthesis phase. Okay, so main function of yes phase when we mention, it is the DNA replication. So DNA replication occurs, and so there is no increase in chromosome number. Okay, so what exactly is happening? So now we know, let's say earlier the chromosome was with only one chromatid, but after the yes phase, so now let's say this is after the yes phase, the chromosome number is not increasing. Okay, so chromosome number is anyway going to remain the same, that is one chromosome here I have shown, so that will remain as a one chromosome. Only thing is that the DNA content is gonna double. Okay, so DNA content which is this, so that is gonna double, so that is the replication. So in that case, so centromere is here, so DNA content, double the content of DNA we can see. So this is one chromosome, so this is also one chromosome because we count the number of chromosomes, so that is based on the number of centromere. So only one centromere is here, so that means only one chromosome is present. Okay, so here also one centromere is here, so that means this is also one chromosome only. So what difference is there? So after the yes phase, you can note that the DNA content has doubled. Okay, so double the amount of DNA we can find, so that is in case of a yes phase. Okay, so no increase in chromosome number, the chromosome number will remain the same. Hi Harsh, good evening. Okay, so initial DNA we could say that the amount was basically 2n, like double the amount of DNA formation you can say. So let's say initially it was 2 chromatids you can find. Okay, so here is one chromatid and here is one chromatid, but final DNA after the synthesis phase, so you can find that there is a double the amount of DNA, so that is 4 chromatids you can find, that means the DNA content is double, so that is 4n. So now initial chromosome number was what? So that is chromosome number was only 1, so that is only one chromosome was there. Even after after the yes phase also, the chromosome number is still the same, that is one chromosome only because the DNA content is double there. Okay, so here in the gap 2 phase, so what exactly is the change that we can see in a gap 2 phase? So now I'll just explain it to you here. So yes phase, these are the changes occurring. Okay, so in growth 2 phase, so before we see that, let's see what exactly happens in a growth 2 phase. So growth 2 you can also call it as gap 2, but since in Selena it is mentioned as growth, so you can go with the growth itself instead of a gap. Okay, so Selena it is mentioned growth, so go with the growth. Okay, so now G2 phase when we mention, so here again the RNA synthesis increases. Okay, and the protein synthesis increases. So what is the cause or what is the reason for this one? Because the DNA is already synthesized. Okay, so now that DNA is going to produce the RNA. Okay, and that RNA is going to produce a protein. So why protein is required? So protein is basically in order to form a plasma membrane, so we require a protein. Okay, and in also in order to form a spindle fiber also. Okay, so spindle fiber formation also requires a protein, so that means tubulin protein is required for a spindle fiber formation. So in that case again we say that the protein requirement is there. So that is with respect to plasma membrane formation and the spindle fiber formation, and that's the reason whatever DNA was synthesized, so that is going to produce a RNA. From RNA only protein synthesis occurs, and this protein is required for plasma membrane. So as the cell division will occur, so that is in the next stage, which is the young phase. So there you can see that the cell division will occur. So daughter cells are being formed, so that means plasma membrane. So there is a furrow formation that you can see. So plasma membrane is being formed. Now in order to form a spindle fiber, so that is that is again made of a tubulin protein. So for that also protein is required. So that's the reason. So RNA protein synthesis, along with that the ATP is also being synthesized. Okay, so ATP is also increasing. So why there is a requirement of ATP? So we can see that the ATP is required, so that is for the cell division to occur, large amount of ATP is required. So that is also being synthesized, and whatever rest of the cell organelles which were not synthesized, so those cell organelles can also be replicated here. Okay, so this is about the G2 phase. So in G2 phase what you can find, that is growth 2 phase, or you can also call it as gap 2 phase, so which is also known as the G2 phase. So synthesis of protein you can find. So as the protein, so basically from DNA only RNA is synthesized, and RNA is the one which is going to produce a protein. And why this protein is required? So that is to form a plasma membrane and a spindle fiber. So synthesis of protein we can see, and overall cell growth can occur. Okay, since all these things are being formed in a cell, like you can see that, so there is a continuous RNA formation, so there is a continuous protein formation. Okay, then DNA is also replicated, so naturally the cell is going to grow. Okay, so cell growth you can find, multiplication of cell organelles you can find, formation of the spindle, so that is the protein required for the spindle for fiber, so that is also kept ready here in the G2 phase. Okay, is this clear? Any doubt till here? Okay, so let's see this one. So YUM phase. Okay, now in YUM phase, so this is the fourth phase which you could see that. So let's say if this is a cell cycle now. Okay, so this is only this much is actually a YUM phase. Okay, so about 95% is actually the interphase only this 5% is the YUM phase that we see. So in that, so this is going to start with the division of the nucleus. So under division of nucleus, so YUM phase, let's write here. So YUM phase occurs in 2 stages. Okay, what are those 2 stages? So one is karyokinesis. Okay, so karyokinesis we can see. The other one is basically a cytokinesis. Okay, so there is a cytokinesis. Now karyokinesis is basically division of nucleus, and cytokinesis is basically a division of cytoplasm. So here, how exactly division of nucleus is going to occur? So this is going to occur in 4 stages. So one is prophase. Okay, so the very first stage is prophase. Then you can see there is a metaphase. Okay, so there is a metaphase. Then you can see there is a anaphase. Okay, so these are the stages that you can find, and then you can find a telophase. So these are the stages. So only after the the karyokinesis is done, so next it will go for the cytokinesis. So where the cell is going to form a furrow like this. Okay, so there you can see a furrow formation. So this furrow is going to get deepened. Okay, so there is a separation of the 2 of the cells that you can find. So there is nuclear membrane which is going to reappeared. So that is in the telophase itself. So nucleolus also has reappeared. So there is a chromatin fiber that you can find. So after the cytokinesis, the cytoplasm is dividing. So here the cytoplasm was there. So before that only the karyokinesis, that means only the nucleus was dividing. Okay, so let's see this one now. M phase. Okay, so karyokinesis occurs in 4 stages. So these are the 4 stages. And in case of animal cells, so cell, what is it? The cytokinesis will occur by the furrow formation. Okay, 1 second. So let's have a look at the cell cycle. So let me just check if this is all. Yeah, fine, right? Okay, so let's see this. Okay, so now when we mention about the cell cycle, okay, so this is G2, or you can see here this one is mentioned as a M phase. So that means now this whole thing what you see phase now, okay, and with respect to that you will start from the G1 phase. Okay, so in phase what you can see, so there is a cell growth. So there is a cell growth, okay, and accumulates essential protein as DNA building block. So basically you can see RNA synthesis, protein synthesis, ATP synthesis, okay, and along with that you can also see that the nucleotides are kept ready which are required for the yes phase. Okay, so that is kept ready, and other than that, so basically all the raw materials in general so are kept ready which are required for the yes phase here. Okay, so next in the yes phase, that is synthesis of DNA, so cell starts to replicate. So that is basically chromosomal DNA. So whatever DNA was there, so that DNA is actually getting replicated. So earlier you can clearly see in this picture. Okay, so now this is one chromosome. Okay, and this is one chromosome. Okay, now you can see here in this phase the chromosome is actually double the amount of DNA is formed in this one. So here the DNA is doubled, and here also the DNA is doubled. Okay, so that means earlier it was only like this. After the yes phase, so DNA replication we can see. So we can see one more strand. So that means the amount of DNA is doubled. So that is only with one common centromere. So here you can find a centromere at this point. Okay, so there is a centromere. So let me just take the pen. Yeah, so here is the centromere. Okay, and cell starts replicating the chromosomal DNA, so results in pair of chromatids. So that is at the centromeric region itself. Now in G2 phase, so that is the growth 2 phase. So final preparation for mitosis, that is the next stage is actually the M phase. So for this the final preparation will be done. So cell is going to provide ATP, so that is large amount of energy production, so that is energy currency production, and synthesize the essential protein. So what are those essential protein? The plasma membrane proteins are the essential protein. The tubulin protein which is required for spindle fiber formation, so that is the essential protein. So all those are being produced here in the G2 phase. Okay, so only after that the M phase will occur, where the cell is going to divide. So that is by 2 phases, the karyokinesis and cytokinesis. So cell nucleus divided, the pair of chromatids will separate from each other. Then in the cytokinesis, the cytoplasm will divide to form 2 daughter cells. So you can see here now one cell which was there, so this itself is getting divided into 2 cells. So this is with respect to animal cells, so that there is a furrow formation that you can see. Okay, so you can see the furrow formation there. Any doubt till here? Is this all clear? Okay, so let's see this part. Okay, So did you know? So one second. Okay, so let's see this part. So now during cancer, so cells will divide and uncontrollably. So basically cancer, when we mention, so uncontrollable cell division. Okay, so this is uncontrollable cell division is basically we can see in a cancerous cell. Okay, so that means usually when a cell is there, so there is a limit. Okay, so depending on the requirement, usually we can see a cell division. But in a cancer cell, because of maybe UV radiation might have caused a cancer, or maybe a particular virus might have caused a cancer. So now based on that, you can see a uncontrollable cell division will occur. So that means continuous cell division will occur, and chemotherapy is done to treat the cancer by killing the cancer cells. So basically chemotherapy is done for those conditions. Now some chemo drugs target different phases of the cell cycle. So now now when we give a chemo drugs, okay, so that is certain drugs are there which can arrest the cell. Okay, so basically arrest the cell. So what is the arresting cell? So arresting cell in the case in the sense, so basically the cell now in G1 phase as well as in yes phase. So yes phase, what is exactly happening? The DNA synthesis occurs there, right? So now as we give a particular drug, so yes phase won't be complete. So yes phase is gonna get arrested. That means there is no DNA replication. So if there is no DNA replication because of the drug that is given or a particular medicine that is given, so in that case we can say that so DNA is not going to replicate, and because of that, so there is no further division of a cell. So that is how we can treat a cancer cell. So that is by arresting the cell at a particular phase of a cell cycle, so that there is no continuity with respect to the cell division, or M phase will not occur. Hi Abhay, good evening. Okay, so let's see this part. Alright, so now gemcitabine is a yes phase drug. So this is a drug which can be taken. So if if a cancer is there, okay, so not at it can be taken only at the early stages. So now this is one of the medicine or a drug which can be taken, and it is going to arrest the cell at the yes phase. So no cell division, no DNA replication. So if no DNA replication, then there is no M phase. So if there is no M phase, so there is basically the number of cells will not increase. So number of cells is gonna remain the same. So there is no cell division in that case. Okay, so we will see this one. So yes phase of the cell cycle, what exactly is the change that we can see? So amount of DNA doubles in each cell. So this is with respect to yes phase. Okay, and amount of DNA remains the same in each cell. Okay, so chromosome number is increased, amount of DNA is reduced to half in each cell. So can anybody give me the answer for this one? Like what exactly might be the answer for this? In yes phase, what is the case that is happening? Any idea? So till now I explained the same thing. Yes, so basically in yes phase you can see that the amount of DNA is gonna double. Okay, so double the amount of DNA production we can see. So during yes or synthesis phase, replication or duplication of chromosomal DNA we can see, and synthesis of histone protein also occurs here in the yes phase itself. So that means some amount of protein is also being produced, and during this time the amount of DNA per cell is gonna double. So now in cell cycle, DNA replication takes place in yes phase itself. Okay, and so that is G1 phase, also called as gap 1 phase or growth 1 phase, is characterized by the increase in cell size And in S phase or synthetic phase, so DNA molecules are going to replicate, and in the G2 phase it is the second growth phase, or we can say gap 2 phase or growth 2 phase, where there is an intensive formation of RNA and protein. So in the mitotic metaphase, the chromosomes are arranged at the equatorial plate. Okay, so all these changes you can see with respect to the DNA. So let's see this one. So given below is a schematic breakup of the phases or stages of a cell cycle. Okay, so you can see here they have mentioned this as the interphase here. Okay, so this one is the interphase and this one is a M phase. Okay, and now what they have mentioned, so this is actually A, and here this is B, this is C, and this is D. Okay, so now you know which of the following is the correct indication of the stage or a phase in the cell cycle. So now C, they are saying that this one is karyokinesis, which is wrong. Okay, so is this C karyokinesis? So C is actually a cytokinesis. Okay, so C is cytokinesis. That means this is wrong now. So what they are asking, so you need to identify the correct one. Okay, then what is mentioned? So D. So D is synthesis phase. So what is mentioned here? So now you know this one is G1 phase, this is S phase, and this is G2 phase. So now you know D is mentioned as a synthesis phase, so which can be correct. And A, they have mentioned this as a cytokinesis, which is absolutely wrong, because this one is a karyokinesis. Okay, so this is a karyokinesis. So in that case, this is wrong now. Okay, and the next one is, what is the B is metaphase. So this is not metaphase, because you can see first stage is prophase, next one is metaphase, and this is anaphase, and this is telophase. So that means this is also wrong. So only correct thing here is the synthetic phase, that is D is the one. Okay, so B is actually anaphase here. So B, we can just write this as a anaphase. Okay, so B is anaphase, not the metaphase. So these three options goes wrong. So only correct one is the option B. Okay, so in schematic breakup of the phases or stages of cell cycle, so D, synthetic phase, is the correct indication of the stage or a phase that is in the cell cycle. Okay, so any doubt till here? So DNA replication in bacteria occurs. So this is, you know, DNA replication in bacteria occurs. So that is within nucleolus. Okay, so whether this occurs within a nucleolus, prior to fission it occurs, or just before the transcription, or during the S phase. So now this is with respect to DNA replication in a bacteria. So do you guys have any idea about this? So where exactly this occurs? So this is prior to fission, or just before the transcription, or during S phase. So now you know this is, you can go with the answer B, that is prior to fission. Okay, so why? Because just before the let's say this is a bacteria now. Okay, so there is a DNA. So this is, let's say, circular DNA is present. Okay, so now you know just before the cell division, so it will form a copy of itself. So prior to the binary fission. Okay, so you can see a furrow formation. So now you can see a bacteria having its own DNA. Okay, so that is a chromosomal DNA. So in that case, so now this has undergone a binary fission. So this is now a binary fission has occurred. Okay, and because of that the daughter cells are being formed. Okay, so this is now a daughter cell, so this is also a daughter cell, so this is a parental cell. Okay, so parent cell formation you can see. So this process is basically a binary fission. So now you know the answer is DNA replication in bacteria occurs prior to the fission. So basically replication will occur. So DNA replication takes place in dash phase of interphase. So it is the S phase of the interphase, or we can also call it as S or a synthesis phase. Okay. Okay, so before I end this session, so let me just say certain, the batches that we have here. Okay, so these batches when we mention, okay, so these batches when we mention, so this is basically, some batches are going to start from May 11th, so some batches are already started. So once you take the subscription, so you will have access to all the batches. Okay. Yes, Ankit, you can tell me what is your doubt. Okay. So for class 10 ICSE Premier League, so that we yesterday on this thing, so that is on YouTube itself, I took one of the IPL. Okay, so that is match one. So in that case, you could see that I discussed about the so cell division and cell cycle. Okay, so all the 3 mark questions, then 5 mark questions are discussed here in this particular IPL. So series of free sessions to cover important topics and questions with Unacademy top educator. So maths, physics, bio and chemistry, so all these subjects are being covered. Okay, so you can find the link in the description. So now there is a gladiators batch, so this is a all subject batch. Okay, so if you are subscribing for class 10, so you will be getting class 11 to 12 subscription absolutely free here. So PCMB is covered, so that is maths, physics, chemistry and biology is covered. The start date is 4th May. Okay, so which is already started, so 2 sessions are done, so you can watch the recordings of these sessions which are already done. So rest of the sessions which are yet to be done, so those you can attend the live sessions. Okay, and the mode of delivery in this batch is English, and there is a Titans batch. Yes, Ankit, what is the doubt? Okay. All right, so let's see this. Okay, so now Titans batch is there. So here this is going to start from May 11th, so you can attend the live sessions that are happening. Okay, again PCMB is covered, so if you are subscribing for class 10, so you will be getting class 11 and 12 subscription also absolutely free. So introducing community, so never miss on any update from your favorite educator. Get session notes and practice material. Share your feedback directly with the educator via poll. Okay, so check out the link to this educator community below in the description box. So now Bucks Bounty, so this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Then report any inappropriate content using the form in the description. Okay, so how you can watch my special classes? All you need to do is download the Unacademy Learner app. So then open the app and click on get started. So once you do that, you can enter your mobile number and OTP. So sign up using your email ID. So you need to you need to add your email ID and your name, other things. Okay, so then you can select the class 6 to 12. Okay, so class 6 to 12 you can select, because there is one more section where you can find competitive exams, so you can go with the class 6 to 12. Okay, then you can choose your category, since you are in ICSE category, so you can choose either the ICSE or ISC category you can choose. And after that, select your preferred language, so you can go for either English or maybe Hindi or English. So depending on preference that you would like to learn in, so you can go for any of these. Then go to the description of this video. Okay, so after you do all this, you can find a link and then click on that. You will get notified. So basically you need to click on and notify me. Okay, so that whenever classes are there, so 10 minutes before the class, so you will get a notification that the so and so educator is going to start a class, so you can join the session. Okay, so you can also use my code. So my code is VaniYT. Okay, so with this code you can unlock the many of the free products that are there on Unacademy. Okay, so you can unlock the free products there. Okay, and other than that, you can also use this code VaniYT to get 10% discount on the subscription. Okay, so this can be done. All right, so see you all in the next session. Okay, so subscribe to our channel, that is Unacademy ICSE and ISC both. Okay, and also click on bell icon to get notified about the sessions. Like and share the video with your friends and download the Unacademy Learner app. This PDF you can join my Telegram channel. So all you need to do is in search section you can search Vani Kari ICSE class 9 and 10. Okay, so this is once Vani Kari ICSE class 9 and 10, you can search on Telegram and you can join my Telegram channel. So all the related notes or any updates regarding the Unacademy, any updates regarding my classes, so I'll be sending it there. Okay, all right then, so see you all in the next session. So any doubts that you guys have? Okay, all right then. Okay, so see you all in the next session. Bye bye everyone, thank you so much, take care.